start. So thank you very much everyone for attending. We had an impressive number of people registered to join us. People are still joining, I can see. The webinar we're doing today is part of a webinar series called Tuesday Lunch with Rita, where we meet on the first Tuesday of each month to discuss specific immunological, rheumatological related topics. I'll start by introducing myself. I'm Despina Eleftheriou, and I'm a professor of pediatric rheumatology, clinically working at Great Ormond Street Hospital and academically based at UCL. And I've been for years interested in research, trying to identify the genetic, I guess, etiology of some of the rare inflammatory diseases we're looking after, and also developing some new therapies. And I'm co-chairing the session with Alexandre, a professor in pediatric rheumatology based at the Hospital Femme-Renfant, who's doing research on early onset autoimmunity. Please remember to ask questions using the chat box or the question function, and we'll go through these at the end. So Alex, over to you.
Okay, thank you very much, Despina, for the introduction, and hi everyone. So we are really delighted to welcome you to this fantastic session. And so today we have Professor Isabelle Meyts, who's a clinician scientist, talented woman responsible of diagnosis and treatment of children affected by primary immunodeficiencies. She is a past president of ECID. She is also talented in research with an ERC grant on data too. And she's also a great person working on different aspects of genetic disease, early onset, and doing bone marrow transplantation. And today she will present her view on how to diagnose these very difficult cases of early onset immune disease, from autoimmune to primary immunodeficiency, all this genetic disease called inborn error of immunity, and she's a specialist of this. She's working in several different networks. So Isabelle, thank you very much for joining, and we're listening to you.
Thank you very much for the invitation and for the overkind introduction, I must say. So I'm going to try and share my screen with you because I think that's working, right? Otherwise you tell me. It's fine? I guess it's fine.
Perfect.
So
thank you.
Actually, when thinking about how to diagnose a primary immunodeficiency, or for the rest of this lecture, actually inborn errors of immunity, I think when we are discussing whether to go for a panel diagnosis or for whole exome sequencing, I could end maybe this talk within a minute, but I think it would be nice to just work with you through a few items that are really important when, even as a clinician, you are using genetic diagnosis that you need to be aware of. And this includes pitfalls, this includes gaps of our knowledge, but it also includes some, let's say, some knowledge that you really need to be able to validate a molecular diagnosis. So of course, if we look at the scenery of inborn errors of immunity, what we are living now is that we have about 500 genetic defects that are described so far, and many more are being described. So in the last 6 months, I can even count almost 50 more that have been described. So it's also true that different variants in a single gene can result in different modes of inheritance and effects of the function of the gene, gain of function or loss of function, which lead to a totally different phenotype in the patient. And so I think we must admit that next generation sequencing has become really the standard for both research and clinical diagnosis. And that's what we have to live with. And that's also, I think, where inborn errors of immunity has really been a nice model, especially when it comes to the validation of the genetic diagnosis. And I think it's key for our patients that we try to achieve a molecular diagnosis, not only for the targeted treatment options that hopefully are available, but also for the counseling of the family. Just to give you the figures here, the increasing numbers of inborn errors of immunity as published in the Journal of Clinical Immunology, and also for this audience, quite importantly, if we look at the recent, the novel immunodeficiency that have been described in the recent update, in fact, most of these are in the innate immunity field and in the autoinflammatory conditions. And we also have to acknowledge that 16 of the 55 novel gene defects published are actually derived from single patient studies, which means that there is a really in-depth validation before you can rely on a single variant in a single patient to be really the determinant of the disease in the patient. I also want to highlight this because, of course, our primary immunodeficiency field was always impregnated by the infection phenotype. And of course, right now we know that a patient can present first maybe with infection, but along the way, all of the other features can come on the travel or let's say the route of the patient with autoimmunity, autoinflammation, malignancy, but also severe allergy. So all of these phenotypes can actually mingle up and really present a complex clinical phenotype. So I think really the way forward is to think of inborn errors of immunity or errors of immunity and really reaching new frontiers with this group of conditions. In fact, in my opinion, every patient presenting to the inborn errors clinic or the PID clinic is really a diagnostic challenge. There is hardly any patients that you can just, follow up and say, I don't really need to know what is the molecular diagnosis, especially in pediatrics, where you really need to know what am I going to see in the future for this child. So any patient with PID also becomes at the same time a research challenge, because there's only a very few cases where you have a known PID phenotype with a known PID mutation, so a pathogenic variant in a known gene. Those are really the, let's say, the minorities. And all of the other patients, you're going to have to delve into genetics and to really combine all the knowledge that you can gather to establish a diagnosis. And now when thinking of the different tools that we have at hand in genetics, it's actually the ideal phenotype would be that we can just sequence a single or a small set of candidate genes. And this could be, for instance, the case for a patient presenting with B- agammaglobulinemia. And if it were a boy, we would all go for BTK sequencing. So we would use Sanger sequencing in this case, which has the, let's say, the advantage of a very low error rate, but of course a very low throughput. So we cannot sequence 500 genes by Sanger sequencing. We could, but it's just a very time-consuming effort and a very expensive effort. So luckily we have now the let's say the advantage of next generation sequencing. And then the crucial question of this seminar came to view. And it means actually, I have a problem with my slides because it's running automatically. So I'm sorry for that. Anyway, if we have next generation sequencing at hand, then it's really important to realize that we can actually sequence a targeted panel, and this can be the entire exome, but it can also be a list of PIG genes that we know of and that we want to look into. So that is really targeting a panel of genes within the bigger exome or next generation sequencing. Of course, we can also choose not to target anything and look for the entire genome, which is the next step. So when we do this, it's important to realize that if you are going for next generation sequencing, what happens is you take the genomic DNA. I'm going to try and put my pointer on, sorry for that. So it's actually that you deconstruct the DNA, or you construct it in a DNA library by various forms. There's some technical steps to repair the ends of the DNA fragments. And then what happens is a massive hybridization step in most of the exome sequencing methodologies. And then you try to map these very short fragments actually along a reference genome. You start to align them so they reconstruct the exomes and the genes. And then you start to call the variants, depending on what you are using. But right now we all know that we are heavily relying on the Broad Institute for calling all these variants. And actually, when you are doing the variant calling, that is when you're reconstructing the differences between the reference genome. And so if you look here, this is whole exome sequencing. You can see the exons of the gene, and there are some hypothetical mutations or pathogenic variants. And you can see that you will not pick all of them up if you just do a whole exome sequencing. You will miss the intronic variants. You will miss the intragenic variants. So you will be stuck with the exonic variants, or maybe the variants in the exon-intron boundaries. I have to say that with the new kits, you can also pick up several of the intronic variants, but these we cannot. It cannot be, let's say, assumed that you will pick up these. Look at the difference in the data that you're getting. So for whole genome sequencing, you're getting about 150 gigabytes, whereas for Wes, you're ending up with a total of 10 gigabytes for the final data that you're working with. So I think this is one of the things to remember. Whole exome sequencing is not the holy grail to find all the interesting variants in your patients, but it is really a useful tool. And we have to also acknowledge that most of the coding, that most of the pathogenic variants are likely to be in the exome coding or the exons of the gene and the protein coding exons. So what I think is really important if you're using, or actually if you're even reading a report that you're receiving from the molecular geneticist, what is really important is that you know that you have to have a certain read depth of the exome. That means that the number of reads per nucleotide that you have need to have a certain level. If you want to, of course, the more the better, but if you want to increase the deep depth, that's going to cost you an amount of money, because you will first need to restrict the number of genes that you want to do, or you're going to have to pay more to have more reads covered. Also, what is important, you need to know what you are targeting, and you need to know exactly, because it may be that some of the genes you are interested in are not even in the targeted region of the capture kit. So that's really important that you know that you have to know some technical details about the kit you are using. And just realize that even with a lower read depth, whole genome sequencing will have a more uniform coverage than whole exome sequencing will do. Even with the newer kits, that is still the case. So we might even say that whole genome sequencing is the next, is probably the future of our diagnostic endeavor. So also, you have to know about the problem with several genes. For instance, we have many genes in the, sorry for this, I don't know how to solve it. If anyone can assist me, I'm happy to do it during the lecture, but it's also important to realize that some of the genes we have in our primary immunodeficiency genes actually have a very high GC content and are therefore very difficult to sequence reliably by next generation sequencing methods. So that's really important to realize. Also, several of the genes have repeat contents, for instance, allorepeats or line repeats, that are very difficult to sequence, and where we have to rely on other techniques to be able to decipher pathogenic variants. And also probably for structures like the telomeres, the centromeres of the genes, it's going to be important to rely on long read sequencing. So that's probably also another frontier that we will have in the next generation sequencing diagnosis. Also very important to know all the pseudogenes. For instance, NEMO has a pseudogene and CF1. So you really need to realize that what you are looking at may be wrong, and that you have to have another alternative diagnosis to really decipher variants in these genes, and not always that easy for some of the genes. Also remember, so that this is it's a bit older paper, but still very nice to realize that whole genome sequencing calls more variants, and the variants they call are likely to be more correct than the variants, some of the variants called by whole exome sequencing. So this is a straightforward comparison between a whole exome sequencing experiment and a whole genome sequencing experiment of several, of an entire cohort. And actually, when you look at the differences, you can see that whole exome sequencing exclusive variants that were called only in the whole exome sequencing were actually more likely to be false than the ones who were called exclusively by whole genome sequencing. And I think that's really important to realize that you can also have wrong calls in exome sequencing, and more so than in whole genome sequencing. Another thing that I would like to discuss with you, and that's really important if you're getting these 20,000 variants as your as your whole exome output, then of course it becomes the key. What is now important for me to focus on for my single patient here? So the allele frequency is quite important. We say that more than 1% allele frequency is actually a common allele, so it's very unlikely to be pathogenic. However, it's also true that several common variants have been described to be pathogenic. For instance, the type 2 variant at this position and the link with tuberculosis, as published by the group of Jean-Laurent Casanova. But also, for instance, CFTR pathogenic variants have a higher allele frequency and are, of course, important for the diagnosis of the patient, which can have a phenotype that is maybe not typical CF, but that is really a lookalike. Also, quite important is that you can think of the functional effect of the variant, although with a lot of caution. So you would assume that variants that are indeed in the exome and that are in, let's say, in a coding position are more important than the ones that are intronic, maybe even more important than synonymous variants, for instance. But actually, we all know that we have all read examples of these variants that are still proven to be pathogenic. So you have to be very careful in dismissing a variant to be pathogenic. And of course, the easiest ones to consider are the nonsense, the stops and the start, the loss of stop, the gain of stop or the loss of start mutations. Those are the more clear ones to consider. But always be careful with the interpretation of any variant. And so I would give it as an advice never to just rely on what the variant looks like. There are some other tools that are really helpful, and I think one of those is for me definitely the PopVis, and I here represent I think my favorite gene, and maybe that's also the favorite gene of Despina. So that is the PopVis of ADA2, and actually what you see here is you see the MAF, so all the mean allele frequencies are given here, and the CAT score, that's a deleteriousness score for the missense variants. And you can actually see per variant that you include, you can actually look at all the data, all the annotations of these variants. And another quite interesting tool is actually to list, if you have a large cohort, you can actually list all the variants that are common to your cohort and that are so above 1% in your cohort, but not found in databases, for instance, publicly available databases like GNOMED. So this means that for your cohort, probably these are due to either technical errors or they're in, for instance, here, as you see, GC-rich regions or ALU regions. And so they are not relevant to pathogenicity, but they can sort of really make things harder if you want to filter down to the causative variant. So I'm sorry for the back and forth all the time, but I hope that we can still make it. So once you're done all of this, look at the quality of the sequencing, etc., then of course it becomes important to realize, so what am I looking at? What is the phenotype of my patient? What does it look like? And you go, for instance, you identify a variant that is of interest, and then you can actually think of this, of using this, the human gene connectome, which actually helps you to prioritize an allele or a gene by biological distance to a lookalike disease maybe, or to a phenotype that you're familiar with. For instance, if you're looking into genes that cause herpes simplex encephalitis, you could look at TOL3 and all the genes related to that, and then maybe compare to your exome results and see if there's any interesting variant popping up that is in one way or another way related to this TOL3 pathway. And this way you can have, for instance, surprises. You can find, for instance, links with genes that you then try to, let's say, delve into from a science point of view. Of course, what is crucial, at least that's what we think now, is that you have to have a gene or a variant in the gene that codes for a protein that is expressed in a cell line or in a tissue that is relevant to the phenotype. And I give this example: IRF7 deficiency, for instance, is quite has been described as a cause of life-threatening influenza A, and in the meantime also for COVID-19. And if you have, of course, West result with 144 variants that are all very rare. But there happens to be only one that is relevant to the pulmonary epithelium and to, for instance, the producers of type 1 interferon, in this case, the plasmacytoid dendritic cells. Then, of course, IRF7 would be the gene to look into. So these are, of course, very easy examples, but if you end up with 20,000 variants, it's not that easy to start prioritizing. And of course, there are some other things that you can use, some data from evolutionary knowledge, and that is whether the gene that you're looking into is under a strong purifying selection. In another, let's say, in another way, the gene damage index needs to be low, which means that this gene doesn't tolerate any changes. That is the ideal situation for a pathogenic variant. And so there's many scores that you can use, but for instance, the residual variation intolerance score, the GDI index, is one of those scores that is really nice to look at. But if, of course, a nucleotide position at a certain position is well conserved, there is a very strong selection, then actually this is an important gene to look at, and this can be really helpful in finding the diagnosis of your patient. I also want to highlight this new prediction score. It's cones. It's supposed to be, and it's probably much better than the state-of-the-art approaches, to look at when you are trying to decipher whether the gene you're looking at, if you only have, for instance, one variant, you need to know, is it possible that this gene would give rise to an autosomal dominant inheritance, or the mutation in this gene would give rise to autosomal dominant inheritance. And for this, you can actually use this score. The lower the score, the stronger selection constraints are acting on the gene. And for instance, this is very low for genes on the X chromosome, but also for genes that have, conditions with a Mendelian inheritance. So that is quite interesting. And there you can see the more negative you go, the more also likely that you have a loss of function, for instance, in these genes. So what is the disadvantage is a score for the gene and not for the individual variants, but there you can go to the, the evolution of the nucleotide position from mammals, all the mammals, and you go all the way to the human beings. Also, what I quite like is actually the combined approach with the mutation significance cutoff. And so I think we all know the CAT score, which is a deleteriousness score published by Yuval Itan and the group of Casanova, and which is really nice because the easy way to look at it is that a score above 15 is actually the cutoff for pathogenicity or likely pathogenicity. But if you combine that with the 95% confidence interval for that given gene, you're actually having a much stronger even predictor of deleteriousness. So for instance, if you have a probably damaging variant from the variant point of view, and you have a gene that is very intolerant to changes, then of course you have the highest chance of working with a variant that is really relevant to the disease. So then I want to say something about our idea of being unbiased. I think we cannot be unbiased when we perform next generation sequencing. I think it's really important to look at the mode of inheritance in the family you are studying, the clinical penetrance, and to be really aware of the fact that the more and more we see incomplete clinical penetrance, and also of several aspects of the disease, it can be different. So you might, for instance, have a patient presenting with rheumatological manifestation as a single patient in the family, but maybe some other patients in the family have thrombocytopenia, etc. So all of these features can have a different degree of penetrance. So that's important. And then, of course, we need to realize that for one phenotype, we can have a dozen of genes and even more. So you have to really be aware of those hypotheses that you're putting forward. And of course, if we're looking from a science point of view, the easiest way is that you have a multi-patient kindred. with, let's say, an autosomal recessive inheritance pattern, and where you have multiple affected individuals that you can study. But of course, we all know that this is rarely the case, and that's so if you're with a single patient in a family that you have to look into the de novos, that it can be really a pain to start looking for the diagnosis. So, and I think this is almost, let's say, the summary, but I think the last line here is the most important. I think experimental validation of a causal relationship is really crucial to a molecular diagnosis. It can be really limiting and rate limiting to really give the diagnosis to the patient, especially if it's a new gene, of course, but also for unknown variants in genes that are well known. And I think that is crucial, and that is something that is really the bottleneck for an optimal molecular diagnosis, in my opinion. So to come, let's say, to an almost conclusion, I would say performing a genetic diagnosis in a patient these days, the way I do it, or the way we do it here, for immunodeficiency and inborn errors of immunity, in fact, in the conditions where we have a patient with a phenotype that I would be able to restrict to 3 or 4 genes, then we would still, if we want to be very fast, we would still go for Sanger sequencing. And I can think of, for instance, a Bruton patient, or especially, let's say, a T minus, B minus, NK plus kit. We have a limited set of genes, and we would go for Sanger. In all other cases, we would go for whole exome sequencing with a panel targeted analysis that we change yearly. And then if we don't find anything there in that restricted panel, or we are not sure of the variants of unknown significance, then we go for the analysis of the entire exome. And I think looking at the pros and cons of everything, of course, it's true there's the risk of incidental findings with whole exome sequencing, but that really is important to communicate to your patients if you're performing this in a research setting, what the expectations are and how you are going to deal with this from an ethical point of view and from a legal point of view as well. And so everything comes in the end down to resources, to a reimbursement for the patient, and especially also the turnaround time. Because if you would be able to do a whole exome sequencing to find a variant in a gene that you already suspect beforehand as a causative gene, then maybe you would even want to go there because of the routine and because of the automation and all the other knowledge you can gather from the, let's say, from the whole exome sequencing. And I think something that we have to be aware of for the future is, of course, the modifier genes, multigenic inheritance, etc., which we will, of course, not uncover if we are going to just do Sanger sequencing for the patients. So I think that's about it for me. Oh, I just wanted to say something about copy number variations, because that is something that even with whole genome sequencing is very difficult to trace down. I'm going to just mention a few that we have tried to use, but not always with great success. You have to have some really nice and consistent NGS data before you can really dive into these platforms or these say pipelines to use. But it's really nice if it works. Exome depth, for instance, if you have a good exome set of your own, and you can really look at the structural variants in your exome sets, which can help you. And otherwise, you also have to be aware that microarray can still help you, and MLPA, of course, where needed. And that's, of course, very important for several genes that are prone to have many CNVs. And then finally, because are we now, with all our fantastic techniques, so really successful in finding the diagnosis for our patients? And actually, that's quite confronting, because in the best, let's say in the best papers, we get up to 60% of diagnosis with an exome panel. In the worst, and that really depends on the constitution of the population as well, in the worst cases, we only get like 30% of diagnosis in the patients that have been receiving whole exome sequencing. So then the question is, what can we do next? And what is the answer? Is it RNA-seq? Is it whole genome sequencing? And there's a few studies trying to tackle this. And let's say that the additional diagnosis is really disappointing. So in these studies, actually, they have 7 additional diagnoses by adding RNA-seq to a panel or to a whole exome sequencing approach. And in this one, they actually have 7.5% diagnostic rate by RNA-seq only. And here in this study in genomic medicine, there's only 7 additional diagnoses after exome sequencing. So that's a bit confronting. So I think we have to be more, I don't know, we'll have to have other techniques and to have more, we have to be more open, I think, for multigenic inheritance. Then I just want to call for this, which I think is really important, but also really the bottleneck these days, because even if we do a gene panel diagnosis, we end up with a list of variants of uncertain significance. And then what do you do with these variants? If you think of a very strong variant, then you still have to end up doing a molecular validation in the lab. So you need to be either collaborating with a lab or you have to build your own lab to do some basic work. But I think collaboration is key because you cannot study 500 genes. It's impossible and nobody's going to fund that. So, but this is really important. You have to prove that there is some functional effect on the pathway, and you have to be able in the best ways to rescue the phenotype, especially if you're the first one to have this variant or to find this variant in a patient. You cannot just call a variant pathogenic. It has 2 big implications for the patients to call a variant pathogenic if it's not proven. And I also want to highlight that published does not equal pathogenic. So there's variants published that we, for instance, thought were pathogenic because they were published, and when we tested them molecularly, we could actually show that it was not pathogenic. And I think in the future, it's going to become very hard to disregard and dismiss a variant of uncertain significance from a result of a gene panel or whole exome sequencing cohort, especially in the clinical work. Okay, in science, you can go for the prioritization of what is interesting for the lab, etc. But in clinic, to really say that this variant is not relevant, even a synonymous variant, is really hard to say. So let's say in practice, depending on the severity of the phenotype of the patient, you can get a yield of 30 to 60%. Of course, also the population structure is important. Addition of RNA-seq or whole genome can push an extra 7 to 10%. But these are the things that we have to be, let's say, thinking of. Multigenic mechanisms, yeah, epigenetic variation. More somatic mutations probably are out there and waiting to be unraveled, like the paper by Megan Cooper on toll-like receptor 8 is very important. So I think if we look in somatic mutations, it becomes even more difficult to assert a diagnosis. And also the question, of course, maybe we don't need to know the gene, maybe we just need to know the pathway to help the patient, and then we can take more time to try and unravel the genetic diagnosis for the counseling aspects. I don't know how to, I think it's doable, and I think there are several omics studies on the way that try to do this, but it's a challenge. And the next thing is, of course, that you can never rely, let's say, on just published data with just the in silico scores of a variant. And I think that's really important as a message here. So I think I've come to the end of this. I hope I didn't, you know, annoy you too much with the automatic play of my slides. And I want to acknowledge all the people in my team, in the lab and in the clinic here, but also especially the colleagues, my trusted colleagues with whom I like to work, and all the PID colleagues, because I think we are blessed with a very good and collaborative community. And I think it's really nice to be able to discuss variants and conditions with colleagues. And scientists should have more lunch breaks, maybe more coffee breaks, but also physicians definitely should have more coffee and lunch breaks to discuss patients and science. And I think that's it for me. Thank you.
Thank you very much, Isabelle. That was great, fantastic. So don't hesitate to ask specific question, and may I can start with the first, and then Despina, we have already prepared several difficult to answer question for you.
Difficult.
No,
That's
no,
not
no,
kind.
difficult. You're an expert. No, I was. You mentioned the somatic mutations, which I don't know. So in pediatric rheumatology, there are a couple of somatic mutation that seems to be important and involving immune dysregulation. And I don't know how much in infectious susceptibility it's frequent or not. And my feeling is not, but maybe you have other feeling. And how can we include this in the strategy? Thinking about somatic mutation, it means it depends on the age, it depends on something else, on the phenotype or whatever.
I don't know what we can include for infectious phenotype. It's true that most of the somatic variants so far are really in rheumatological disease or autoimmune disease or yeah, pre-malignant predisposition. I don't have a good answer, Alexandre. I think indeed with age we have more risk of having that. On the other hand, I think we might also miss I think early in life, there's also several somatic variants that can happen and that can evolve over time. But then the question becomes, can we pick it up with soon enough to have a relevant explanation for it? We have to put the cutoffs. We have to learn about that. I actually cannot give a good answer there. I don't know if you have a better answer, but I actually don't know.
No, I think we are just starting to discover this
Yeah.
sort of
Yeah, and also thanks to the
variant.
techniques, of course, huh? Yeah, I don't know.
And I guess if I was gonna ask something along the same lines, thinking about the different technologies, Isabelle, in terms of whole exome sequencing, whole genome sequencing, targeted panel sequencing, is there an advantage to one over the other for detecting somatic mosaicism?
Well, I'm guessing that that whole genome sequencing there is going to help you because you will have better reads. You will have, for instance, we were chasing one time we were chasing a somatic variant. We thought it was a somatic variant in a whole exome sequencing report, and then when we ended up looking closer, it was just a technical error. So you need to have the best reliable sequencing technique, and I think these days that's going to be whole genome sequencing. And then of course you need to also preemptively start sampling your patients. Yeah, I mean at several time points you have to have different tissues available. I mean it becomes really complex to do that with the the number 150 patients who present at your clinic to do all this biobanking. Yes, it's doable, but it's extremely expensive as well. Yeah, you need to have the best technique.
So for that, you need a depth. So it means that in panel, it's easier to catch up the somatic variant instead of maybe whole genome, which is not in terms of depth.
Well,
it's not
yeah,
so
but the
much.
quality is better. That's the thing. So even if you have less reads at a single position, the quality seems to be better. And so that's why you might, even if you think that you have enough reads or more reads with your targeted panel, if it's exome based, you can still be wrong. Especially if you use the exome to use the target on. But if you have just a targeted panel and only sequence those, then yes, then you would have higher reads for a single gene. But if you use the exome to decipher the targeted panel, then you lose the benefit. If that's what you mean. I couldn't hear you well.
I think we have some question in the Q&A. So I can start with in limited resource countries where genetic is not available, what is your recommendation in patients who present with combined autoimmunity, allergy, other not fitting specific diagnosis? So if you can't come up with the final diagnosis in the absence of genetic screening, what's the best regarding especially bone marrow transplantation perspective? Have you some advice?
Well, first of all, I think there's in the community there's many physicians and scientists that want to help, and that would, in a collaborative, mostly scientific endeavor, try to find the diagnosis for your patients. And those, of course, are mostly the big labs because they have more funding available. On the other hand, I think there's also the Jeffrey Model Foundation possibility, who have a sequencing program that is free for the patients. I think I would go trying to do that. And then if you are totally stuck with a patient and it's a very severe condition where you need quite some immunosuppressants, no, then we go back to maybe when we didn't have those tools available and we would transplant the patient without having the diagnosis. Yeah. But of course, that's not what we want to advertise, but you know, reality is there and we cannot ignore it. But I would say find a strong collaborative lab and try to work with them. That's doable, I think.
We have additional question I see. So how to handle pseudogene problem from Satish Kumar Loganathan.
Yeah. So for the pseudogenes, it depends a little bit. So for instance, for NCF1, you can have really, let's say, adapted sequencing methods where you use Sanger sequencing to do this single gene. For NEMO, it becomes even more difficult, but there's been a few published methods that are really able to decipher variants in the exact gene and not in the pseudogene. So it's a bit of a gene per gene case, as you just have to be aware that what you're looking at can be in the pseudogene. So it depends on the gene.
I see another question. I don't know, Despina, if you can see the question. I think there were a technical issue for Despina, but still, Bodo Grimbacher is asking, great talk, Isabelle. Don't be afraid by the question, but it's a challenging one. So thank you very much. You said you have proven a published variant to be non-pathogenic. You have to. So, however, how can you be sure that your functional test did detect the impairment conferred by the variant?
That's a tricky question. But if you have like a gene defect, and it's not an uncommon gene defect, it's not an uncommon error of immunity, and if it's a gain, you have to have some gain, right? Mm-hmm. And if you do it, and it was published as, you know, validated pathogenic variants, and if you use the machinery that everybody is using to validate the pathogenicity, and it gives nothing, then I would say it's not pathogenic with, let's say, 99% certainty. I hope that pleases Bodo, but Bodo is much better geneticist than I am. I'm just using genetics.
So still, for someone who uses genetics, there is another question from Antonios Gontaras. In patients with persistent hypogammaglobulinemia after rituximab therapy, is there a role for NGS diagnosis, to your opinion?
Yeah, of course, definitely. I don't believe in coincidences. So if you have a patient not recovering B cells after rituximab, there's definitely something to look for, because yeah, our bone marrow is quite potent, and to not, you know, exit any more B cells anymore, that's really odd. And there's many cases described where they find in the end an underlying inborn error of immunity.
Another question that comes from Shahad Al-Ansari. When should we repeat whole exome sequencing in case we have negative results, but we still suspect to have an error of immunity?
Well, I think when there's big leaps in the technologies, in the kits that are provided by the companies, then I would redo it. And that means, of course, actually I would not redo exome sequencing, then I would go for whole genome sequencing right now. But there's definitely been major improvements from 2000, let's say 14, 15 until now. It's a totally different field now. Like, for instance, a lot of the knowledge on sequencing exons and missing variants, etc. With gaps in the exome kits, it's all been very much improved, of course, now. But still, I think whole genome sequencing is then the way to go to. On the other hand, what you can do if you have the exome available is to, of course, reanalyze every year a new targeted panel. Because if you have every new year, you have 100 more genes, and yeah, you have some more hits.
I don't see any additional questions. So Despina, maybe you have some question for Isabelle.
Yeah, I guess I was going to ask about the variants of unknown significance, Isabelle. So in the UK, a lot of, not research, but actual clinical genetics labs have taken the approach of not reporting these variants. So thinking that perhaps what today is a variant of unknown significance that might become a class 4 or 5 pathogenic variant in a year's time, what's been your approach and what's your thinking about reporting back that this is?
Yeah, so of course I'm not in charge of the molecular diagnostics here in immunodeficiency, but with the team in the routine diagnostics, we've actually come up with a strategy to have in the official report only, you know, the already described variants. Yes. But for us as clinicians, the list of variants of uncertain significance available, which is really very helpful because we should list it up. In several cases, we found by reading, by digging into these variants, really found the diagnosis. For instance, in the mutations in domains that had not been reported to be linked to pathogenicity yet. Unexpected phenotypes, so for instance, like the TREF3 initially reported in herpes simplex encephalitis, but then you find it in several patients with hypergamma and autoimmunity. Yeah, now there's the papers, so that becomes helpful. So if you don't list them up, you lose the information, and that's really sad because it's so much of, I mean, resources gone to waste.
Absolutely. And I guess would the message for our audience be that if they strongly still suspect, despite a negative result, a genetic component to the presentation of the patient, they should still have a conversation, dialogue, and rescreen?
Yeah. Yes. Yeah. And of course, it all means money. But on the other hand, if you find a diagnosis, it's also the stop of the, let's say, the odyssey of the patients. No doctor shopping, no expensive treatments that won't work, etc., etc. So I'm a believer in the key of diagnostics. So yeah.
I have an additional question from the chat. So how do you anticipate the nanopore long reads will do in the field of immunology? So the long read sequencing that managed to catch several genes in a row. So what's your opinion in that? It's been useful in oncology. What in immunology?
Well, yeah, I think it definitely has a place. If it's going to conquer the entire field, I don't know, but it definitely has a place to. We should have actually a straightforward comparison with this technique and find what it can bring as an addition to what we already have with the regular short read sequencing. Actually, I don't know. Probably people in the audience know, but we've seen at the genetics working party some really nice results, indeed, mostly from oncology, but definitely it's going to have applications in immunodeficiency as well. And also for some of the structural variations, it can really help. So yes, there will be definitely a place. I just don't know how we would integrate that for the moment.
Yeah, money is the main issue probably still.
Yeah, as usual.
Okay. Is there any burning question in the audience? If not, I don't see any new question. I think that's okay. We come to the end of the talk and the meeting. So I would like to warmly thank you, Isabelle, for this discussion talk, and thank Despina for chairing the session with me. So thank you everyone for participating to the webinar. You also welcome to join us on the next Tuesday. It will be June the 6th to learn about autoimmune encephalitis, normally, with the Professor Honora. The registration link will be sent as usual, so don't forget to record you. And also I should mention that you can still listen to any of the webinar available on the website. And I wish you a good afternoon and see you. And thank you to everyone. Bye-bye.
Thank you, everyone.
Bye.
Bye.
Bye-bye.